In this video, we're going to learn about protein synthesis. Protein synthesis is the process of making protein. There are two steps involved in protein synthesis: transcription and translation. Transcription happens inside of the nucleus, while translation happens at the ribosome. Let's learn about the language of DNA. For example, in English, we have 26 letters that we can use to make words. But in the language of DNA, they only have 4 letters used to make words. For example, in English, we would call this a word, and that word is made up of what we call letters. But in the language of DNA, this is called a codon, and a codon is made up of nucleotides, or also called nitrogen bases. In English, a word can be made of many letters, but in DNA, a codon is only made of 3 nucleotides. Now let's learn about the base pair rule. In DNA, A always pairs with T, adenine pairs with thymine, and C pairs with G. Cytosine pairs with guanine. So an easy trick to remember is apples in a tree, A pairs with T. Car in a garage, C pairs with G. Transcription happens inside of the nucleus. And all these little stringy pieces are the DNA. And what happens is that DNA separates from each other. The two strands separate. It kind of unzips. And then it creates a new strand called RNA. And now for translation. That newly produced RNA leaves the nucleus and becomes the messenger RNA. These groups of 3 are the codons, or the words, as we would call it in English. A codon is a group of 3 nucleic acids. This structure right here is called transfer RNA, or tRNA for short. It's made up of the anticodons, which are a group of 3 nucleotides that pair up with the codons of the messenger RNA. And this at the top is called the amino acid. That's what builds the protein. So now let's find the amino acid. So we're going to use this. So we're going to find the amino acid by using a codon wheel. So we can break this up into 4 sections. So let's look at the anticodons, AGA. AGA would pair up with U, C, and A pairs with U, because we're working with RNA now, not DNA. So in this case, A pairs with U, and G still always pairs with C. So now we find these codons, UCU, and the codon wheel. So we start off with the inner, and we work our way to the outer. So we're going to find the U, and there it is. Now let's look for the C. It's right there. And now for the U, right there, and the amino acid is serine. Now let's see another example. The anticodons is UUC. So which codons do you think this matches with? If you say AAG, you are correct. So now let's find the AAG on our codon wheel. So we start inwards and we work our way outwards. So we find our A right there. We find the other A, which is right there. and we find the G, which is right there, and the amino acid is lysine. So now let's transcribe a messenger RNA. So if the DNA strand is TAC, then it should pair with A, U, because we're working with RNA now, and G. T pairs with A, C pairs with G, G pairs with C, T pairs with A, T pairs with A, C pairs with G, G pairs with C, A pairs with U, T pairs with A, G pairs with C, and A pairs with U. So now we find the amino acid. So we start off with the A, right there. We find the U, which is right there. And we find the G, which is right here. And the amino acid is MET, which is the beginning of the sequence. Now let's find A, G, and C. So there's our A, there's the G, and there's the C. And the amino acid is serine. Now let's find AAG. There's A, there's the other A, and there's the G. The amino acid, lysine. Now for CCU. There's the C, there's the other C, there's the U. Proline is the amino acid. Now let's find UU and U. There's the U, there's the other U, and there's the third U. And PHE is the amino acid. Now for ACU. There's the A, there's the C, there's the U, and THR is the amino acid. Congratulations, you just made a protein. I hope you found this video helpful, and thank you for watching.